Good afternoon, everyone, and welcome to the series of agronomy seminars, semester spring 2022. It is my pleasure to introduce to you Professor Gori Johal, who is a professor of maize genetics and pathology in the Department of Botany and Plant Pathology here at Purdue. Dr. Johal works around two main research. One is the mechanisms of corn immunity, and the second one is genes that are involved in plant architecture. In the second research, Dr. Johal cloned several dwarfing genes, including AIR1, NANA1, and BRACTIC2, and his latest discovery is a dominant dwarfing gene named D16, which I'm sure everyone is interested to hear more about it. And then with that, without any further ado, I will let Dr. Johal to take a stage and continue the talk.
Okay, great. Well, thanks a lot for the kind introduction, and I'm very happy to be presenting the seminar. The title is Short-stature corn, a short-lived fad or the corn of the future.
Guri, I think you need to share the slides.
All right.
You see the word.
This is amazing, right?
Yeah.
Yeah, so I must say that the title of the talk was kind of encouraged by a YouTube video that I was watching on short-stature corn. And when I was watching this particular video by Don Joplin from Maxi Seed, he said, is it really the corn of the future, or is it really one of those fads that people have been talking about?
I'm sorry to interrupt.
That's how I got the title of the seminar.
Your screen is not shared still.
Okay, I have organized my talk into three different themes. The first one is the history and background of dwarfing as an agriculture trade. We are still working on the technology, sorry. Okay, so in three different themes, and the first one, the history and background of the dwarfing trade of dwarfing as an agriculture trade, and why corn breeders failed to harness it when everybody else, rice breeders, wheat breeders, barley breeders, they took great advantage of that to enhance yields. And lastly, why there is a certain interest in this trade, and what's the best way forward? So whenever we talk about plant dwarfing, the first name that comes to mind is Norman Borlaug. So these two things are kind of synonymous, and rightfully so, because it was both the scientific as well as humanitarian efforts of Norman that led to the introduction of dwarfing varieties of wheat in Mexico as well as in the Indian continent, and that changed the lives of many people. It made India within a few years from a food insecure country to food secure country. Not only actually India, but also Pakistan and a little bit Bangladesh as well. So, and the dwarfing gene, or the genes that he was working with, they came from the variety Noren tamed, a variety which was developed in Japan and released in 1935. But its previous history is not very clear. So probably it was already there, but in 1935 it was I think released over a much larger area. And of course, it was recognized during the Second World War, and it was brought to the US by perhaps Orville Vogel at Washington State University. and he released many varieties using this dwarfing gene. Norman 10 seems to have two different dwarfing loci, one on genome B and the other one on genome D. As you all probably realize, that wheat is a hexaploid plant. It has three different genomes, A, B, and D. So this particular gene, let me see. Yeah, this particular gene called RHT1, abbreviation of reduced height. It's a dominant trait, and as mentioned, there are two of those, one on chromosome B and of the genome, and the other one on chromosome D of the genome. And there were parallel developments in rice at the International Rice Research Institute. And they used a dwarfing gene that came from the variety DG Wujen from China, which translates into, I had actually written something here and it somehow got translated into Chinese, and Mosen removed that. But that means that it's a short-legged and brown-tipped. DG Wujen means in Chinese.
Gurmukh, sorry to interrupt.
Short-legged and brown-tipped.
Gurmukh, sorry to interrupt.
Anyway, the first variety that was developed by using this DG Wujen as one of the parents was IR8, and it was released to many countries in South Asia and the Orient, and it enhanced the yields by 3 or 4-fold, basically within a few years.
Guri, we can't see your
Was
slides.
done by
Guri,
Beechel and Gurdev Kush. And both of them got the World Food Prize, I think,
can't see your
in the
slide.
90s for their work. And this particular dwarfing gene from DG Wujen is a single recessive mutant gene, and it has been designated as SD1, semi-dwarf. But I was surprised to learn that it's the lowly sorghum, which actually was the first one where the dwarfing trait was harnessed as a commercial trait. And seems like it was in 1855, a dwarf variety of sorghum was released to farmers, both in Ohio and Penn State in 1855. And as mentioned, it came to me as a surprise. And a lot of genetic studies were initiated quite early as well. And by the 1940s, efforts by Carper and Quimby, it led to the identification of 4 different dwarfing genes, which they designated DW1, DW2, DW3, and DW4. And they also used these 4 different dwarfing mutations in various combinations to produce very short plants as 4-gene dwarfs, 3-gene dwarfs, as well as 2-gene dwarfs as well. And the dwarfing gene that received special attention from them was DW3, even though it was.
There is a problem with the slides. Excuse me, guys. We have a little bit of technology problem here. The slides are not moving. Yeah. Yeah. Are you here?
A couple more. going up, more up, more up. Yeah, more up. Yeah, so yeah, that's yeah.
I hope this time things are okay.
Mohsen, now
Can we
it's
give you
good.
feedback?
Mohsen.
Sorry about that. I think we realized that
The slides are not moving.
you guys were not able to see my slides very well. But Mohsen, who's been helping me, I'm in his actually office, so he hopefully he has fixed the problem. Okay, as I mentioned, sorghum was actually the first crop species where the dwarfing trait was initially characterized and commercialized as well. And also the genetic studies were done in this crop species much earlier compared to everywhere else. And as early as by the 1940s, efforts of Carper and Quimby at Texas A&M University, it led to the identification of 4 different dwarfing genes in sorghum. And these guys actually were able to use these 4 dwarfing genes or loci to produce dwarfing varieties ranging in height from very small to medium size. So they were able to produce very short dwarfs containing combinations of 4 dwarfing mutations or various combinations where they had 3 different gene dwarfs or 2 different gene dwarfs. And during this time, they also realized that one of the mutations which they designate DW3, dwarf 3, it was very important for the harvest index of the plant, even though it was genetically unstable mutation because it was reverting back to the wild type at different rates. It was one of the mutants that they prefer to have it in their cultivars. Most of the dwarfing traits that have been utilized so far to reduce the height of crop plants, they are either due to defects in the biosynthesis or the action of various plant hormones. And the one that comes at the top of the list is the gibberellic acid or GAs. In the case of the rice SOD1 gene, it was the biosynthesis of GAs that was compromised and which led to the semi-dwarf mutant phenotype or dwarfing phenotype. And in the case of RHD1 of wheat, these mutants are defective in the action or signaling of GA. So even though GA is produced, but it cannot really cause the plants to grow tall. Brassicas steroids, which are very similar to testosterone in mammals. They're also another class of phytohormones, which can impact plant height and growth. And in the case of mutants where biosynthesis of BRs is impaired in dwarfing mutants, we have one case in maize NANA1 mutants. And in the case of barley Uzu gene mutant, which has been actually commercialized, although NANA1 has not been commercialized so far, but we are hopeful that maybe one day it will be. The Uzu gene in barley, which has been commercialized, it is defective in the action of brassicas steroids. It's a mutation in the receptor of brassicas steroid. So there's really no action, even though there's a lot of brassicas steroids. And the third class of phytohormones which are involved in controlling plant height are auxins, but it's not their synthesis or action that seems to be involved in controlling plant height, at least so far, but it's their distribution, because auxins are produced at the top of the plant and then they're transported in a polar fashion from the top to other parts of the plant, to the bottom and the roots. And that is somehow impaired in the maize Burkitic 2 and the sorghum dwarf 3 mutants. And this is the sorghum dwarf mutant that I mentioned earlier, which Carper and Kimby like quite a bit because they enhance the harvest index of the plant quite a bit. So as I mentioned earlier, GA biosynthesis seems to be the most important pathway involving cell elongation. So I'm going to spend a few more minutes on this one because in the case of those genes which were involved in green revolution, both used by Norman Borlaug and the breeders at Erie, they both have defects in the GA biology, GA metabolism, so to say. So this is the pathway which starts in the plastid, and in the plastid starts with GGDP and converts that into chlorine, which then leaves the plastid and hangs around in the endoplasmic reticulum, eventually producing this compound GA12 with the action of these different enzymes. And GA12 is then either converted directly by these enzymes, at least these are all encoded by the same gene, GA20 oxidase, to different forms, and eventually produce these various bioactive GAs, GA5, GA6, GA3, GA1, GA4, and GA7. But GA12 can also be converted by GA13 oxidase into GA53, and then that gets converted into these as well. So both GA20 oxidase and GA3 oxidase, they're the GA biosynthetic enzyme. But of course, we don't want to have too much GA hanging around. So as long as their function is done, then these are actually broken down or inactivated by GA2 oxidase, which just like GA3 oxidase or GA20 oxidase can act on a bunch of different compounds or intermediates. There are hundreds of these GA compounds or GA intermediates. Some of them are bioactive, but mostly of them are not active. So I think I'm just going to show, so this is GA12 is very important. That's the first, I guess, really important GA molecule that is produced with the action of many of these enzymes. And then it moves into the cytosol, where it gets converted either into GA4, GA7, or by the action of GA13 oxidase, it gets converted to GA53 and into the 13 hydroxylation pathway. And these are, this GA20 oxidase is involved in that. And so are these GA3 oxidase, all encoded by one gene, and they can act on different molecules of the GA pathway. These are all biosynthetic enzymes, and they lead to the production of these bioactive molecules. As I mentioned earlier, GA2 oxidase, it can inactivate GA active molecules or their intermediates, inactivate them. Okay, so so much of that work was done in Arabidopsis, but we have many of these mutants in this biosynthetic pathway available in corn as well. And one of them is called anther-1, AN1, that is defective in the very first step from GGDP to CPS. And in the case of dwarf 5 or D5, the defect is from CPS to corine here. The D3 mutants are defective in GA13 oxidase, and the D1 mutants are defective in GA3 oxidase. We haven't identified any maize mutants which are defective in this one, but you can actually generate those by, perhaps there's a functional redundancy, there are probably 2 genes at least, but you can actually transgenically inactivate it by either RNAi or some other means. And then there are two other mutants in cone as well called D8 and D9, which are dominant mutants, and they're in the Gala protein. And I'm going to come to that next. So once this GA is produced, then what does it do next to cause cell elongation or mediate plant growth? Sorry, before I get into that, so in the case of corn, GA mutants, they look like this. So they more fit like ornamental cone plant than what can be used in the field because they have broad leaves and stay very short. And they also have another phenotype, which is anthers in the ear. So when GAs are deficient or they cannot be sensed by the plant, then the anthers appear in the ear. But if the GAs are there, then these anthers do not produce as in this wild-type plant. So GAs are considered as feminizing hormones in corn. In contrast, brassinosteroids are the masculinizing hormones, as we showed with this Nana-1 plant a few years ago. So when BRs are defective, then ears are normal, but in the anther, we have seed. It's called tassel seed phenotype. And the antherian phenotype looks like this if you have the ear fully pollinated. So the action of GAs is controlled by this protein, Gala protein. And Gala protein is, it has a number of different domains. And Gala is the domain here and the carboxy terminal. And Gala is simply, I guess, first a few amino acids are DE, LLA, and then something else. So that's where it derives its name from the sequence of the amino acids in this protein. So people divide this part of the protein into Gala domain, which is GA response specific. And then the second half of the molecule is called GRAS domain, which actually is the function domain. It also has the nucleus localization signal on it as well. So these Gala proteins, they keep all the transcription factors or transcription regulators under check when there's no GA. So if there's no GA, all these genes which are involved in elongation, they are not able to act because of the Gala protein, which acts as a repressor. And this Gala is able to sequester these transcription factors or transcriptional regulators and preventing them from triggering these genes which are involved in GA responsive processes, cell elongation, and stuff like that. So if there's no GA, Galas keep acting as a repressor, preventing these transcription factors from triggering these genes and keeping the plant short. However, in the presence of GA, gibberellic acid, the receptor of GA is GID1, which was initially identified in Arabidopsis. I guess it's GA insensitive dwarf. That's the name of the gene. So GID1 is both in the cytoplasm as well as the nucleus, and Gala is mostly in the nucleus. So in the presence of GA, which binds with GID1, either in cytoplasm or in the nucleus, and their interaction causes them to interact with Gala. And this interaction recruits a ligase, which is able to, add ubiquitin molecules onto it, which is a marker for degradation of the protein by this proteasome machinery. So in the presence of GA, Gala is targeted for degradation. And when it gets degraded, these transcription factors are let loose. They are let free. They're able to then trigger these GA responsive genes, which brings about elongation. So in the absence of GA, plants stay short, but when the GA is there, the plants are normal in height. So this is basically I guess the basic biology of the dwarfing trait in plants. Again, here I have only mentioned about GA, but same goes for brassicas, steroids, and others as well. So as I mentioned earlier, dwarfing was used as a trait to enhance crop performance for everything out there, for wheat, for rice, for sorghum, barley, everything. But it could not be harnessed in the case of corn. So the question is why? I looked into that, and there seemed like there were many factors which kind of conspired together to prevent the exploitation of the dwarfing trait or the dwarfing mutants in the case of corn. The first one is the architecture of the maize plant. Maize plant is very different than many of the other plants because there are two components of height in maize. First is the overall height of the plant, which includes everything plus the tassel, right? And the second is the height of the ear. So and the height of the ear actually puts a kind of constraint on what can be done with dwarfing mutation. If the height of the ear is too low, then it's not acceptable to breeders or to agronomists, because then they have to change the harvesting machinery or equipment. And that was one reason why it probably, and there were no mutants at the time where the height of the ear was still decent and the height of the overall plant was relatively short. And the mutant availability, as I said, there wasn't really any mutation which fulfilled both of these criteria, relatively short overall, but the ear still high enough so that it could be used for breeding. As I mentioned earlier, there were many mutants available, both in the GA biosynthetic pathway and the GA signaling pathway, but most were really severe or null, and none of those could be really exploited for reducing the height of the maize plant. The genetic background effect is also very important, and for some reason maize is just obnoxious for that. Oftentimes mutants behave very unpredictably in different backgrounds. So in one background, they may seem just fine, but in another one, they will be either too severe or too suppressed. And that's one reason why the first mutant of the GA biosynthetic pathway is not called a dwarf mutant, but N3R1. Because the dwarf phenotype of that N3R1 mutant is not very stable. It is there sometimes, but other times it's not there. However, its other phenotype, which is the feminization of the ear, is there. And that's why it was called N3R1 and not dwarf 1 or dwarf whatever. Another reason is the maize stem. It's not as worthless as that of wheat and rice, both of which are hollow and relatively very weak stems. Maize stem is relatively strong, and it can be easily, I guess, stiffened by having different kinds of genes, which can impact the structure of the cell wall, the lignin, or whatever. And that's actually people will do by using stiff stalk germplasm that was generated. And the hybrid nature of the cone crop is another factor that mitigate against the dwarfing trait. So in the case to make a hybrid, you have to have both a female parent as well as a male parent. And if the mutation that you're trying to use to create dwarf cone is a recessive mutation, it has to be introgressed into both the male and the female parents. But when you put a dwarfing mutation in the female parent, it compromises vigor sometimes so much that that producing a hybrid seed with that becomes cost inhibitive. And it's become similar to what the problem with the first generation of inbreds. These first generation of inbreds are so weak that people could not make hybrid seed by making simply single crosses between two inbred lines of two different heterotic groups. They actually had to first generate hybrids between two inbreds in the same heterotic group. And then these two hybrids from different heterotic groups were crossed together in a double cross hybrid to produce a hybrid seed. So the same problem comes in our way when the vigor of the female plant gets relatively compromised or weak when we introduce a dwarfing mutation into it. This is just to show how hybrids are produced in the beginning when male geneticists or breeders realized that actually you can generate inbred lines and then produce hybrid. But these lines were so weak, they actually had to cross two lines or the same heterotic group A and B to produce a hybrid, C and D to produce another hybrid, and then cross them together to produce the double cross hybrid. It worked, but it's not very convenient and it's not as good as a single hybrid. A single hybrid is when you have one parent cross with second parent directly to produce F1 hybrid. And it became possible for the first time in 1950s when maize geneticists and breeders were able to develop inbreds which were robust enough to produce enough seed for hybrid seed industry. But I think that the fact that was really responsible for, I guess, putting a nail in the coffin for dwarfing technology in cone was the fact that cone yield in cone could be enhanced by another trick as well. And that was by enhancing the density of planting in cone. So this slide here shows the increase in yield of corn from 1860s all the way to 2010s or 2020s. And as you can see, there was no increase in yield during the open pollinated cultivars era up until 1940. And that's when the double cross hybrids were introduced and people start seeing an increase in yield. And then in about 1950s, single cross hybrids were introduced. And since then, there has been a linear increase in yield, for the last 60 or so years. But much of this increase in yield, it came from an increase in the population density of the plants in the field. So on this side, it's corn yield, and it was increasing from 1900s to 2010s. Much of it came from an increase of population density from 10,000 around 1950s to about 35,000 in 2010s or so. So that was probably one reason why, because corn breeders were able to get similar increases that people got with the green revolution genes, with increasing the density of the plant. So corn breeders almost forgot about the dwarfing trait. Why need a dwarfing trait when you can increase yields other ways? But then it brings us to the next question: why there has been so much interest lately And much of this interest has been, I guess, initiated by efforts at Bayer and Bayer, and they have been very vocal about it, saying that, you know, next big thing in corn is actually a dwarf corn, or that's the future of corn. And so the question is, you know, what are the reasons behind that? Again, one can think of many different reasons, and one of them is the environmental urgency. And again, Bayer has been using it in their PR, saying that short hybrids are fairly resistant to strong winds, and that's true as well. But they are not really resistant to Dracos, and sometimes they say that. But regardless, yeah, short hybrids actually are very resilient, especially if the stem is sturdy, and the standability of the crop is much better than if the plants are tall. And we also have more knowledge about the genes which are responsible for many of these dwarfing traits as well. And having this knowledge, there's always this possibility we can actually somehow manipulate them by either genetic means or transgenic means. And of course, gene editing has become very important as well. And many of the companies are relying on gene editing as being the next approach in plant breeding. But still, I think the biggest reason is the biggest reason for going for short corn is that that if we have to increase yields further, we have to increase the density still. With the current architecture, the tall plant, which is 9 to 10 feet tall, I think we have reached the limit, or almost limit, and with this current architecture, it's going to be very hard to push the plant density further from 40,000 or so. Okay. The only way we can do it, and people accept that, is if we change the architecture of the plant. And that's probably one reason why many of the companies are trying to do that. Because if you can enhance the density, planting density, you actually can sell more seed to farmers. So if you take a look at how many seeds are there in a bag of corn when it's sold to farmers, there are 80,000 seeds. And if you can plant 2 acres with it, 40,000 each, and that's much less compared to if you can plant 60,000 per acre. So that's where I guess much of the margin is for the companies. And that's why they're trying to push for higher densities. And to be able to do so, they have to change the architecture of the plant, and that comes from having a dwarfing trait. And this is another graph to show the same thing as well, that we are reaching a ceiling with regard to how far we can go with planting density per acre before running into problems. If you go beyond 30,000 or 45,000, whatever, the plants start suffering from lodging and other problems. Okay. And I think there's consensus that if you want to go any further, we have to change the architecture of the plant. And one way to do that is by having plants or hybrids which are relatively shorter. And one of the slogans that Bayer has been using, they're calling that if you use short-stature hybrids, they will pay much better. And in their, I guess, the terminology, pay stands for protection against winds and storms, access to the crop throughout the growing season. What they mean by that is that if your crop is relatively short, then you can mount all sorts of equipment on your tractor to spray fertilizers and pesticides as you need to do that. And of course, you can enhance yield by having short crops as well by enhancing planting densities. So this is a slogan from Bayer. But again, I think as good as sound, I think there are problems, and some of those problems are, again, as I mentioned earlier, two things that will dictate whether a dwarfing trait will succeed or not. And those two points are that if a mutant trait, a mutant gene is impacted by the genetic background, that it is very severe in one background and relatively expressed in another, it's not going to succeed. And we have been working on these for the last 20 years or so. It was in our lab that we cloned the BRCA2 gene of corn, showing that it actually is involved in the polar movement of auxins from the top of the plant to the bottom. And we also showed that the equivalent gene and the mutation in sorghum was DW3, which was unstable, even though it was appreciated by the breeders a lot. So we have seen that many of these mutations, including BRCA2, that they're easily impacted by the genetic background. And that makes the trait relatively unpredictable, or you have to pay special attention to it, how it behaves in different genetic backgrounds. Another factor you have to keep in mind is that the phenotype cannot be too severe or not too weak. If it's too severe, it's going to impact the yield of the hybrid seed from the female plant, and that's going to come in the way of cost effectiveness. But if the phenotype is too weak, then it's easily masked by having the hybrid vigor in the hybrid plant. And again, as mentioned, we have very severe leaves of BRCA2, and we have fairly weak leaves of BRCA2, and we haven't been able to produce a very good hybrid because of these problems using BRCA2. So because of genetic background, yeah, and that's one actually huge reason why it is relatively hard to tame the BRCA or the dwarfing trait in corn. And in my opinion, what actually we need is a dominant trait. A dominant trait, of course, which is not impacted too much by the genetic background and is not too severe or not too weak. And if you have genetic dominant trait which can reduce hybrid trait by at least 2 feet, and it can be propagated as a homozygote, that is the way to go. That's the way to actually introduce the dwarfing trait in corn. There seems to be a very narrow window of opportunity. If the dwarfing trait is too severe, it becomes cost prohibitive. If the dwarfing trait is relatively weak, then it basically disappears and you cannot really use it very effectively. So you need a mutant which is just right, just like the Goldilocks mutant. And that has been a challenge. Nobody has been able to identify a mutant like that. But I think we have maybe hit a lottery, a jackpot. We have found a mutant which follows all of these criteria fairly closely. We have it's dominant mutant. We have designated it D16, because D15 up to D15 mutants have already been identified or reported in corn. D16 is semi-dwarf mutant, which reduces the height of the corn hybrid in heterozygous condition by about 2 to 3 feet. So in this picture, we have hybrid plants of MOS17 and B73, a hybrid that was very popular in 1960s and early 70s. It's one of the most heterotic hybrid. But if you put D16 in this hybrid background in one dose, then that's what happens to the hybrid. It's about 2 to 3 feet shorter. The plant is sturdier. The size of the ear is very similar to what we have in the wild type plant, and it seems otherwise it seems very almost identical to that of the wild type. We actually introgressed that, or we made hybrid of this D16 in the B73 background with a number of other inbred lines as well, both from the ex-PVP germplasm, as well as some of the local or the earlier inbreds. And we have seen this difference between the wild type and the mutant hybrid about 2 to 3 feet. And it seems like the genetic background has minimal impact on this trait. So these two criteria, that the hybrid is 2 to 3 feet shorter and it's not really impacted too much by the genetic background, they're both satisfied by this D16 mutant trait. We generated this by EMS mutagenesis. And again, as mentioned, this in the B73 background. We have not cloned the gene yet, so we don't know what the gene is. And that's something we are doing right now. And we're hopeful that we'll have the gene cloned in the next 2 to 3 months or so. But I think this mutant has the potential to transform the entire corn industry for years to come, and has a big potential to bring a lot of royalty to Purdue in the process. And I think with that, I will stop and entertain any questions that you may have. Thanks.
Yeah, it is a good time. Thank you very much, everyone, for listening, and sorry for the technical difficulties that we had. Now we open up a question for you guys. If you have any question for Dr. Johal, please go ahead.
I have one question. So Gauri, this is great that you found this new variety.
Any
So,
question for Dr. Johal?
Hello?
I think so.
Mohsen, can you hear me?
Yes, can we?
You may have to type the question in the chat for
Yes,
them
sir.
to see it.
We cannot hear it, no. Okay, so please go ahead. Please go ahead and type your questions in the chat box.
So we can hear.
We can't hear from them. It's strange. Oh, thank you. It's also not difficult here. So what we do is here. Corey, you need to go through these things.
Mhm.
Okay. This is one of the questions.
Okay, so yeah, there's one question in the chat box that if you have a dwarfing trait in the hybrid, that is going to bring the ear very close to the soil level. And will that, in addition to impacting, of course, the ability to harvest it with with the machinery, will it also make it more susceptible to diseases because it will raise the humidity level and may also enhance soil-borne pathogens as well? No, definitely. So again, yeah, we cannot bring it. I guess the minimum height of ear from the ground has to be at least 3 feet. Although at there they're saying 2 feet will do it, but I think in my opinion it has to be at least 3 feet or so. So you're correct. Yeah, bringing the height of the ear way close to the ground will have many unintended problems or unrecognized problems. Yeah, that is correct.
Mohsen?
Yeah, I think we can hear from you guys now. If you want to ask your questions, just turn on your mic and ask.
Yeah, we can hear now. So so go ahead if you have any questions, please.
So Guri, I have a question. This new variety that you described, is it less shading? Does it have a favorable canopy architecture? I mean, to prevent shading and the loss of productivity?
Yeah, those are some of the things we haven't really done yet in a lot of detail. But the architecture of the plant looks very similar to the wild-type plant, except that it is relatively short. And it has, maybe a little more upright leaves compared to the wild-type plant, but not, again, there's not much difference. So we think that unlike the Burkitic 2 trait, I think, which is shown here, I don't think we will have that many problems with D16 so far. And it does not bring the ear very low. The compression of the internodes is kind of uniform throughout, as opposed to Burkitic 2, which compresses the internodes more below the ear compared to nodes on the top. And that's what one of the biggest problem is with Burkitic 2. If you try to increase the height of the Burkitic 2 from the ground, then you actually make the plant pretty tall on top as well. So, and there's no easy fix for that. But in the case of D16, we don't have that problem at all. We saw that in the inbred, we saw that in all the hybrids that we made as well.
This question in the chat box.
Well, how does it compare with the current bare short cone hybrids? We haven't seen those, but at Bayer, as much as I can tell, they're using 2 different genes to bring the height of their plants down. One of them is Burkitic 2, and the Burkitic 2 gene was cloned in our lab way back in 2003, and we know all the pros and cons of the Burkitic 2 gene and the mutants derived from that. So again, Burkitic 2 was recognized as probably the best dwarfing trait in corn way back in 1960s and probably early 70s, but it still was not could not be used as a commercial dwarfing trait.
You have Misha's question.
So again, we having worked with both Burkitic 2 and with D16, I think Burkitic 2 doesn't come very close to D16. Our trait is much better, and because Burkitic 2 is recessive and D16 is dominant, D16 can be introgressed into only one of the parents, the male, and that also allows people to produce hybrid seed fairly effectively as well.
Question from Mitch.
So yeah, the question from Mitch saying Burkitic 2 has pleiotropic effects on tassel size or panicle size. and stay green. Does D16 have any other effects on plant growth or development? Yeah, that's a very good question. We did notice a little bit impact on the width of the leaves, which was slightly enhanced in some hybrids. And I think Jay Hilbert noticed that as well, but it wasn't there in all the hybrids. But other than that, there's very little impact on some impact on maybe stay green, but not too much. And the plants look very similar to the wild type hybrid with regard to rate of growth, with regard to maturity level, tassel size, and everything.
From Tony again.
Yeah, question from Tony. The bare short cone hybrids have larger ear sizes going into the R1 period. And the bare short hybrids have higher leaf nitrogen. So there are multiple traits that impact yield in short hybrids beyond height alone. That's actually nice to know, but we don't really have any of these data with the D16 so far. As I mentioned, the leaves are slightly greener, perhaps compared to the wild type inbred, and maybe slightly more stay green, I guess, ability as well. But when we look at the hybrids between, and the difference is actually, there's very little difference between the wild type plant and the D16 hybrid in many of the genetic backgrounds that we tested. And the only difference is between the height of the plants.
So other questions for Gori?
Hey, Guri, I have one more question. This is Mitch.
Yeah, hi, Mitch.
Some plant breeders and agronomists have suggested that the GA biosynthesis or perception mutants that are used in rice and wheat may have some negative baggage associated with them. Any thoughts about the potential application of some of these, a new mutation that has a phenotype like you've described for D16 for some of these other cereal crops?
Yeah, that's a very good question. And actually people have realized, so I mentioned that it was Orville Vogel who first introduced Norrin 10 into the US, but he was working at Washington State University. And in that area, you have to plant your wheat fairly deep, as much as 6 inches in the ground. And the dwarfing varieties did not survive that. And the one reason for that is these genes are expressed, they control the size of the coleoptile or mesocotyl, as we call it in corn. And when you have those mutations, either Norrin 10, I mean RHT or ST1, then the size of the coleoptile or mesocotyl is compromised and stays small. So it has difficulty pushing the seedling outside the soil. And people in Australia are especially concerned about that because of problems with drought and so on and so forth. And they consider that these green revolution dwarfing genes are actually a problem for them. And they are actually trying to breed their germplasm away from it and looking for other mutations which do not impact the size of the coleoptile or the mesocotyl. Burkitic 2 is like that in corn, and this D8 does not impact the coleoptile size at all either. Okay. Yeah, but that's, yeah. So I think in wheat they have probably identified one trait, one gene, but I'm not sure if they have cloned the gene yet or not.
Other question for Gori? All right. So if there are no further questions for Gori, please join me. Thanks, Gori. for the very nice presentation. And thank you all for attending seminar today. And that was great.
Thanks, yeah.
Thank you very
Bye-bye.
much.
Yeah.